Okay, so let's see. Uh, today is the 29th, which is right here. Yes, so your chapter 5 quiz was due about 15 minutes ago. Some people have been asking, yeah, I will accept these late. I'll take off like 10%. I mean, if it's really late, maybe 20%, but I will accept these late. So go ahead and do them even if you haven't done it yet. Um, You also have a Master and his Emissary quiz, which is, I believe, due Thursday at 9:15 a.m. Originally on the syllabus, I was going to make these due the night before, but I've just found myself being more and more lenient with these, so I'm usually just making them due right before class at 9:15 a.m. So yeah, those are today. We're going to continue talking about Chapter 5, which is sensation, um, and you have reading quiz due next time. Okay. So here's where we left off. We left off talking about the auditory system. And that's kind of at the beginning of this chapter. What we're going to do is talk about each sensation, each of the five senses in isolation, kind of zoom in on each sensory system. Um, and we went through the auditory system last time. And then next, I think we have the visual system, and then we'll go through smell and taste and touch, I think in that order, but might be a little off on the order there. So we'll go through each sensory system by itself. Then we'll kind of come back towards the end of the semester and we'll start going towards the perceptual side of things, particularly with respect to the visual system, because that's the system where there's probably the most known about perception side of things. We'll also talk about synesthesia and multimodal perception towards the end of the chapter. So moving on to the visual system, well, I guess I want to quickly show you in the auditory system, just to remind you, we kind of had these different, we talked about how what we're actually sensing in the auditory system is waves, vibrating air molecules. And the properties of those waves, like amplitude, wavelength, and then timbre, which we didn't talk about too much, but those map on to basically different aspects of what you actually hear. Amplitude, basically the height of the wave, corresponds to loudness, and wavelength, basically the distance between two peaks, corresponds to the perception of pitch. And basically, a big wavelength is a low pitch, and a small wavelength is a high pitched sound. And we have a similar type of deal in the visual system. There's waves that are being sensed by your visual system, by receptors in the visual system, and what you see is influenced by the properties of those waves. And so here, those properties, which do map on in an analogous sense to the properties that I just showed about in the auditory system, uh, hue, or which is kind of like frequency or pitch in the auditory system. But in the visual system, what's known as hue, which is related to the wavelength, that basically is color. Hue basically means color. And it's basically similarly though, it's like the wavelength basically. If you have a very long wavelength like this, you tend to see the color as red. It's on the red side of the spectrum. And if you have a really short wavelength like this, you perceive it on the blue purple side of the spectrum. So purple's way at the end, or they might call it violet or indigo or whatever. But basically, this is the blue side of the spectrum. And if it's a short wavelength, it's over here. This is the red side of the spectrum. If it's a big wavelength, it'll be over here. So that maps on the analogous property of waves in the auditory system would be wavelength, which corresponds in the auditory system to how the frequency or the pitch that you hear. Brightness in the visual system is kind of like amplitude in the auditory system. It's the height of the wave. So in the auditory system, when you have a wave that has a big amplitude or is tall, you perceive it as loud. In the visual system, when you have a light wave that is tall, you perceive it as more bright. So kind of analogous terms would be intensity or contrast, which can be shown right here. So at the top of this little chart, these would be letters that have strong contrast relative to the background. Dark letters have very strong contrast relative to the background, but then when you get down here, the contrast is very minimal. I can't even really, maybe I need an eye exam. I can barely tell with, actually I can see what those are. Now that I focus, I can see what those letters are at the bottom pretty okay. So this would be related to differences in brightness or contrast. A lot of contrast here, very little contrast here. And contrast and or that is brightness has to do with the amplitude or the height of the wave right here. And then saturation slash purity, this is kind of like timbre. And in the auditory system, timbre is the idea that you can have the note C that is played by a piano, and you can also have the note C played by a guitar. And those two notes will have the same frequency, the same pitch, but they'll still have different textures or a different feel when you hear them. Most people can tell the difference between a guitar playing the note C and a piano playing the note C, or a violin playing the note C. Even though they're all playing the same note, each instrument has its own unique sound, its own unique texture, its own unique timbre. And an analogous thing in the visual system is saturation slash purity, which I'm not going to go into the details of what what the properties of the waves that produce this are, but I will say that saturation is the idea that you can have like mixtures of colors. So you can have different shades of blue or different shades of any color, and that's saturation. Basically the way that the different frequency or the different colors, the different hues combine is saturation, in a similar way to the way the different the properties of the waves that produce different timbres in the auditory system. Okay, so now we're looking here at the anatomy of the eye. So this is where visual information from outside in the world, the electromagnetic radiation or the light waves from out in the world, come into contact with your body for the first time, and transduction occurs. That is, the electromagnetic radiation is transformed into a pattern of electrical activity and action potentials, which are then sent through the optic nerve to the brain. So that all that, the transduction process, sensation and transduction begin right here in the retina. So the retina is this part of the back of the eye. I'm not going to require you to know all the anatomy up here. I do want you to know that obviously this is the front of the eye. This is the inside of the eye. And towards the back of the eye, this whole like back half of the eyeball is the retina. And this place is important because this is where the two major types of visual receptor neurons or visual receptor cells are. And those two types, you've probably heard of these before. Cones and rods are the two types of visual receptors which is where visual transduction begins, where electromagnetic radiation begins its transformation into the currency of the nervous system, that is action potentials and electrical fluctuations. So a couple things about the retina. Once again, this is where the rods and cones are, but they're not homogeneously distributed throughout the retina. Cones, which are a type of neural receptor or type of visual receptor, which are important, particularly when it's right outside. And they're important for seeing like the details of the world. At least that's one way to think about what they do. They give you color information, and they also give you, when you're looking at something directly and you're fixating that thing, looking straight at something, what you're doing is you're taking the light from out in the world that's bouncing off the object. For instance, I'm looking at my sunglasses right here and I'm looking directly at them. Basically what I'm doing is I'm taking the light which is bouncing off of my sunglasses, and I'm directing that light directly onto a specific part of my retina known as the fovea, which is basically the center part of my fovea. And the fovea has a lot of cones. So I mentioned rods and cones are not equally distributed throughout the retina. It turns out that most of the cones, in the book reinforces this idea, most of the cones are in the center of the retina, in the place called the fovea. The fovea is basically the center of the retina where the cones are most dense. There's a lot of cones on the center of the retina. And as I mentioned, which when you're looking directly at something, you're taking the light from that something and shining it directly on your cones. And presumably it is your cones that allow you to see the detailed information. So I'm looking at my glasses and there's this little diamond, little metal thing right there. And when I look at it really close, I can see that whoever painted this got a little bit of paint on that diamond thing. So the glasses are white around that little diamond thing. But I can see that whoever painted these got a little bit of a smudge of paint on that little metal metallic diamond looking thing right there. And that's, that required acuity, visual acuity, and my access to the visual details of the world. And I do it by taking that light, shining it directly on my fovea, which is where my cones are, which are allowing me to see detailed information about the world. Contrasting what the cones do and where they're located with the rods don't really give you much information about color of things. And they also don't give you a lot of information about details. It's usually suggested that rods give you information about the gist, the big picture. And they do that in particular by giving you information about contrast and brightness. They're particularly good at detecting when there's contrast. And remember, contrast is this. So rods are pretty good at telling when there's a dark something next to a light something. Like as right here, there's dark letters, next to light letters. And rods are good at detecting contrast. It says dark next to light right here, which is an important element of getting the gist of seeing. I don't know exactly what, like they're particularly good, rods are, when something is in your peripheral vision. And when something in your peripheral vision, you don't really see the details of the thing, but if something's in your peripheral vision, you can tell that something's there, but you don't necessarily see the details of that something. And that's because you're using your rods, and rods give you information about, general information about what's over here, like gist, and they do so by using basic mechanisms like contrast. Is there a dark thing next to a light thing? I might not be able to tell exactly what it is, but I can tell that there's a dark thing next to a light thing. And I mentioned that I've implied that rods are used primarily, they're important when when it's like if you're in a dark environment. And they're also important when information is presented in your peripheral vision. And the reason this is because the rods are densest on the peripheral parts of your retina. So once again, this whole thing at the back is the retina. When you're directly fixating something, the light is going onto the center of the retina, that is the fovea. But when something is in your peripheral vision, like if it's in your peripheral vision, that information is no longer going to the center of your retina. That is, it's no longer going to the fovea. It's going to one of the places surrounding the fovea. Those places surrounding the fovea are where most of the rods hang out. The rods do not give you detailed information. They give you basic information like contrast, which helps you kind of identify the gist, the big picture as to what's going on out in the world. So these are differences between cones and rods. The book goes into more detail about cones than I'm going to go into detail about cones here in the lecture. I do want to say this just again, because this is an important part, and these are nice pictures to supplement this idea. Here's two different pictures. This whole thing right here is the retina. This should actually say receptors on the retina. In fact, I'm going to change it right now. Receptors on the retina. This whole thing right here is the retina. In the center is the fovea, and over here is the peripheral parts of the retina. And so this is kind of a weird dependent variable here, but this is basically, if it's on the red side, that means you have a lot of rods in that particular area. If it's on the blue side, that means you have a lot of cones. So here in the center of the retina, that is the fovea. In this picture, you can see that you have lots of cones. And then on the peripheral side, peripheral parts of things, you can see that there's more rods on the peripheral. And this is taken from a different textbook that I'm borrowing from. The fovea is the center part, and you can see that there's a lot of cones. There are some rods in the fovea, but most of the rods are in the peripheral part, and then cones are very dense in the fovea, which is the center of the retina. So once again, I'll just say this kind of to get the main point here. Cones are densest in the center of the retina. That location is the fovea, where our rods are relatively more dense on the peripheral parts of the retina. So a little bit more about the path of once transduction begins, there's this idea that the information, the electromagnetic radiation from out in the world is transduced, that is, it's turned into some pattern of electrical impulses in the nervous system. And basically what we're going to be talking about here for the next couple slides is the pathway that electrical the electrical signals travel from initially from the eye and eventually until they get to the brain. And so there's a lot going on here. These are actually very similar pictures. Once again, I don't care tons about the anatomy of the eye. I do want you to know that the retina is back here. The fovea is at the center of the retina. I want you to know which photoreceptors are densest on the fovea relative to the peripheral parts of the retina. That's what I want you to know about the anatomy of the eye. I do want you to note here is the optic nerve. So it's adjacent to the retina. And the optic nerve is where information starts heading to the brain. So here what we're doing, so I do want to draw a little bit of attention to this, which is a close-up of some micro stuff, which is going on here in the retina. So you can see it says right here, back of the retina. So this is the location towards the back of the eye. And this is where the rods and cones are, and identifies them right here. Rods and cones. This is the back of the retina. It's a close-up of one part of the retina. There are a good amount of cones here, so this is probably pretty close to the fovea because there are a lot of cones. There's oh wait, I take that back. There are more rods here, so this appears to be in the peripheral part. There's not that many cones, there's more rods, so this appears to be in the peripheral part of the retina rather than the fovea. But here are rods and cones. These are the sensory receptors for the visual system. And this picture and this one as well illustrates a concept which is talked about in the textbook, which they talk about convergence and divergence in the textbook. I'm more interested in us knowing the concept of convergence. So that's the thing that I'm going to focus in on a lecture is convergence. Convergence is the idea that initially when sensory receptors pick up visual information from out in the world, there's a lot of visual receptors. There's many rods and cones that are taking in that visual information from out in the world. But then that information, once transduction begins, once the electrical signals begin, that information is kind of compressed. That is, all that information, which is initially represented by many rods and cones, is then sent to a smaller amount of cells. And I actually don't care if we know the names of these cells. So I'm not going to require you to know that these are called bipolar cells. These are called ganglion cells. There are also things called horizontal cells. Those are all in there. But I want you to understand that the idea of convergence is the idea that you have initially many receptors, but that information from those receptors is condensed, or it converges to a smaller amount of neurons right here, different types of neurons. These are basically relay neurons in the sense that they're taking information and they're sending it along, which will eventually reach the optic nerve right here. But it's convergence because many cells, many different types of many receptors right here are taking that information and it's being compressed down to a smaller amount of cells right here. And that's what convergence is. Taking something which is represented by many things and compressing that information so that it's represented by a smaller amount of things. That is the idea of convergence. And it seems to be a theme in the brain, where the initial steps oftentimes are very detailed, lots of receptors at the very initial stages of sensation. But then as the nervous system moves away from sensation more towards the perceptual side of things, the details are omitted and convergence occurs. Less amount of cells are representing things as we get further away from the initial stages of sensation and transduction. This is showing basically the same thing. Here are the receptor cells. Yeah, this is the one where there's lots of cones. So these pink things here are the cones, the blue purplish things are the rods. So this one looks to be more like it's coming from the fovea, because whereas this picture has a lot of rods in it, which would be the peripheral parts of the retina, this one has relatively more cones in it. So this is probably closer to the fovea right here. But once again, you can see convergence. We have many different types of many receptors right here, and that information represented in the receptors is compressed. It's sent to a smaller amount of cells, and then those cells eventually will relay the information to the optic nerve, which is right here, and then you might be able to guess that the optic nerve takes that information to the brain, which is what we're talking about right here. So here's some interesting things. I believe we've probably talked about this, and certainly you have had, if you've been keeping up with the master and his emissary readings, this is something which has been talked about in the master and his emissary readings, this idea that information in each eye, some of the information travels to the contralateral hemisphere and some of the information travels to the ipsilateral hemisphere. The really crucial determinant of whether information travels to the opposite hemisphere, that is contralateral, or ipsilateral hemisphere, meaning the same hemisphere, is whether the information, the thing you're looking at, is presented in the left visual field, in which case, no matter which eye it is, if it's in the left visual field, then that information tends to travel to your right hemisphere. And you can see it right here. So left visual field is right here. This is the left eye. That information hits the right side of the eye and then travels to the right hemisphere. Follow the red pathway here. Here is the left information in the left visual field. It once again will hit the right side of the right eye, and that information will travel to the right hemisphere. So it doesn't really matter which eye is being struck by the light. What actually matters is what side of the world is that information on. If it's on the left side of the world, it's going to go primarily, the information is going to go primarily to the right hemisphere. If the information is in the right side of the world, that is the right visual field, then that information goes primarily to the left hemisphere. Right side of the world goes primarily to the left hemisphere. So this is what is meant by primarily a contralateral projections. Information on the right visual field goes to the contralateral hemisphere, that is the left hemisphere, and same with information in the left visual field, it goes to the contralateral hemisphere, that is the right hemisphere. From there, there's this thing right here called optic chiasm, which means optic crossing. As far as I understand, chiasm just means crossing, and you can see that there's a crossing right here. What this means is some of the signals from the left eye will cross and go over to the right hemisphere, and some of the signals from the right eye will cross and go over to the left hemisphere. From here, there's kind of a branching into two different pathways. The primary pathway goes to the lateral geniculate nucleus of the thalamus. And this was discussed in your textbook. These are two pretty well-known pathways in the visual system. Most of the neurons from your eye will go to the lateral geniculate nucleus of the thalamus. And we've talked about this idea that all of the senses except for smell will go to the thalamus before it reaches the primary sensory area for that sense. And the visual system is definitely one of the ones that goes through the thalamus. It does so at the lateral geniculate nucleus, which is shown here, and then there would be one on the right hemisphere as well. Then the information from the thalamus, that is the lateral geniculate nucleus, passes on to primary visual cortex, which is right here. Anyone remember what lobe of the brain primary visual cortex is in? You remember that?
Occipital?
It is the occipital lobe, which is at the back of the brain. So then most of the neurons I mentioned go from the eye to the thalamus to the primary visual cortex. There is another pathway. Some of the information from the eye actually first goes to the superior colliculus, which is in the brainstem midbrain region. You can see how it's physically a little bit lower. It's down further in the brain. And so I've heard estimates like 90% of the neurons or 80% of the neurons go to the thalamus, and then like 10 or 20% of the neurons go to the superior colliculus. And presumably different information is processed in each of these different pathways. So very early on in the visual system, though, there's like this fork in the road. Some of the visual information is going to the thalamus, some of it's going to the superior colliculus, and they're processing different aspects of the visual world. The one that the most amount of research has been on, and the one we'll talk about the most, is the pathway involving primary visual cortex and the thalamus, and that is the lateral geniculate nucleus of the thalamus. Yeah, so this is a nice illustration of what's going on in primary visual cortex, or at least the most studied phenomenon, I would say, in primary visual cortex is this, which has come primarily from studies in animals like cats and in monkeys, where they'll stick electrodes in adjacent cells in primary visual cortex. Once again, we're in the occipital lobe back here, and you stick electrodes in adjacent cells. So in one trial, you'll stick an electrode into the primary visual cortex right there, and then you'll present different stimuli, and you'll see the electrical response of the neurons which are close to this electrode right here. And these stimuli you would show in this study are lines of different orientations, so very simple stimuli. The basic question is here is, how does the neural response of this electrode and primary, of this set of neurons which are touching this electrode and primary visual cortex, how does that neuron or neurons respond to lines of different angles, of different orientations like this? So you put an electrode in one place in primary visual cortex, present those different pictures, and you measure the amount of neural responses at that electrode. Then you move the electrode to a new adjacent place in primary visual cortex, and you do the same thing. You present those pictures again, the lines of different orientation. You measure the electrical activity of the neuron or neurons touching this electrode, and you continue on going through kind of millimeter by millimeter of primary visual cortex, which is what this is right here, millimeter track distance. So this is going through basically all the primary visual cortex with an electrode in each different place, and you're presenting visual images to the animal that the animal is actually looking at. And you're looking at the neural responses of each of these neurons, depending on where the electrode is placed. And what they find is that different locations in primary visual cortex respond to lines of different orientations. So this is just the location that you are along primary visual cortex. So starting on one side of primary visual cortex and moving to the other side. And what they show is that right here, the neurons that are touching this electrode, that looks to be around, I don't even know, it's like 20 or 30 degrees. And then, I don't completely understand how to read this. And then it's moving up, and it's like right here, these neurons are 0 degrees. These neurons right here are responding to 30 degrees. These are 60 degrees, 90 degrees, so on and so forth. And so you can see there's kind of a continuum where as you move from left to the right, the neurons are responding to line angles that are kind of continuous. The more you move to the right, there's kind of this shifting of what would be known as the preferred orientation. The preferred orientation is the orientation of a stimulus which causes a given neuron or a set of adjacent neurons to fire the most amount of action potentials. So whatever neurons are right here, these neurons are responding most strongly to around -60. So an angle which is, presumably that's -60 right there. A response of -60, that would be the preferred direction of the neurons, which are located right here in primary visual cortex, because there a lot of action potentials occur when the stimulus which is being presented to the animal is -60 degrees. So the main feature, just to be very clear, that has been studied in primary visual cortex, and there's a lot of evidence that primary visual cortex is dedicated to processing differences in orientation of lines. So a very basic, simple feature. And to kind of connect this back, primary auditory cortex is most known for representing differences in pitch, that is difference in frequency. And primary visual cortex, on the other hand, is most known for representing differences in the orientation of lines. Here's a video. I'm not going to show this. It's kind of hard to show videos, and this is like a 10-minute video. I do suggest watching this because this is some of the actual research. It's old now. It's like from the 70s or maybe late 60s even. But this is basically is the very research which was used, some of the first research that was used to discover this idea that different cells in primary visual cortex respond to lines of different orientations. And it's very, very clear in the video. They give lots of examples. They actually show you, show a study where an animal has electrodes hooked up to its visual cortex, and then light lines of different orientation pass in front of the eyes of the animal. And then they have the electrode hooked up to an amplifier. So when action potentials occur, you can hear the action potentials occurring. And basically for a given neuron, if its preferred direction is a line like this, when a line like this is passed in front of the animal, you'll hear lots of action potentials. But if a line that's vertical goes in front of the animal, the given neuron will not fire action potentials, because the neurons in primary visual cortex each have a preferred direction. They have a direction of a line that they fire a lot of action potentials for. But if the line is not of the preferred direction, then the neuron does not fire a lot of action potentials. Each neuron kind of has its preferred orientation, its preferred direction, and it only fires action potentials when its preferred orientation is shown, or at least something that's very close to its preferred orientation is shown. Okay, so that's vision. We will come back to vision. Vision, and I've mentioned this before, vision is out of all five senses, vision is probably the one, I would say not even probably, it's pretty unambiguously the one where there's been the most amount of research on it. And we will come back. Some of the more interesting things that occur are in perception are known through demonstrations of vision. And so we'll come back to that towards the end of the semester, or the end of the class, and we will also talk about audio-visual interactions, that is multimodal perception. Okay, so now smell or olfaction. This is the sense that does not go through the thalamus. So here, though, there is kind of analogous processes to what we've talked about, especially the visual system. We've already talked about convergence. And you can see convergence right here. Here are olfactory receptor cells right here. These things are kind of analogous to like rods and cones in the visual system. They're the things that they take in information that goes in your nose, and that information for smell is not necessarily a wave like it was for auditory, for sound and for sight. In sound and sight, you have waves, and in olfaction, the thing which you're processing isn't waves, or it's not radiation or energy. In olfaction, it's chemicals that you're processing. I think it says it right here. Yeah, the receptors in olfaction respond to different chemical properties. So chemistry presumably is really important for understanding the subjective experience of smell that you have. Each of these receptors is basically processing the chemistry or the chemical properties of the things which get into your nose. And then that's how the subjective experience of smell presumably is basically your nervous system processing the chemical properties of the things which are entering your body and through your nose. So there are a lot of types of receptors. Probably not all of the types of smell receptors have even been discovered or classified yet. So we'll just say, we'll leave it at the fact that there are a lot of different receptors in the smell system. Each one processes different chemical properties. And then that information is sent along, and you can see convergence here because we have many different receptors. And that information that is representing smells from many different receptors is then condensed. These things are glomeruli, that's the plural, which is basically another type of relay neuron or a relay cell. It takes that information from the receptors, it condenses it, kind of compresses the information before it sends it via the olfactory nerve towards the brain. Yeah, that's really all I want to talk about here. So here are the receptors once again. Here are the glomeruli. Once again, here is the olfactory nerve, which is taking that information. You can see the compression right here quite nicely. Lots of different receptors sending their signals to a smaller amount of neurons right here before they're sent over to the brain. So you can see that right there. But then there is the pathway from the olfactory nerve, which would be somewhere in here. That information eventually finds its way to primary olfactory cortex, which here it's shown right here. So I remember, you recall at the beginning of the class, I mentioned that this is, I will not require you to know specifically where primary olfactory cortex is. And part of the reason is it's located on the boundary between the frontal and temporal lobes. And so here on this picture, it looks like they're showing us the frontal lobe, but really the primary olfactory cortex is kind of, if you took the temporal lobe and the frontal lobe and pulled it apart, it's kind of wedged in between the frontal and temporal lobes. Here it's shown right here. And then, so this is the primary sensory area. We'll talk a little bit about this in a second. I think I had a question about this for your quiz for today. And then that information is sent to what's known as secondary olfactory cortex, also known as orbitofrontal cortex right here. And we'll talk a little bit about the functions of these two different areas here. So just a couple basic things about olfaction before we talk about the brain and what the different parts of the primary and secondary olfactory cortex are doing for smell. I've already mentioned olfaction. Smell does not go through the thalamus. I don't really understand or know what the implications of this. I don't completely understand why some senses go through the thalamus and why smell doesn't. And I don't know what the implications are for this as far as how smell might be different from the other senses because it doesn't go through the thalamus. I guess one possibility is that going through the thalamus is somehow related to the fact that a lot of information in the other senses will cross to the contralateral hemisphere, whereas smell does not. There's no contralateral. It's not like your left nostril goes to the right hemisphere and your right nostril goes to the left hemisphere. There does not appear to be anything like that for smell. And so that may or may not be related to the fact that smell does not, that's just a speculation, that smell does not go through the thalamus, therefore it might not cross to the contralateral hemisphere. Smell tends to stay primarily in the same hemisphere, that is the ipsilateral hemisphere. Another thing about smell, though, is that it's highly connected to limbic regions involved in emotion and motivation. Now, this is, I guess, to kind of get the idea that sometimes old feelings can be stimulated in an individual by smelling things that you haven't smelled in a long time. Smells can remind you of people, and they can remind you of places that you have strong feelings or emotions for. So there is strong emotional connections between the sense of smell and, the evocative emotional memories. But once again, the idea that these things occur because the sense of smell does not travel to the thalamus is a mere speculation. So these are isolated facts. It's hard to know if these things are all interrelated to each other. So once again, I did tell you that we'd be talking about primary smell cortex, and then we talk a little bit about, so one of the things, like, yes, if you look here, olfactory cortex, these are both pictures that are taken from your textbook. Here, olfactory cortex appears like it's in the temporal lobe. But right here, primary olfactory cortex, to me, this looks like it's kind of in the frontal lobe. So like the reason why they're doing this is because, once again, really primary olfactory cortex is located in the wedge between the frontal and temporal lobes. And so, but it's hard to represent that in a picture. And so presumably the way they do it is that sometimes they show it kind of here in the temporal lobe, and sometimes they'll show it over here in the frontal lobe, but really it's in between the frontal and the temporal lobe. So here's a study that they talk about in your textbook. And the thing that's probably most counterintuitive about primary olfactory cortex is its main function does not appear to be to actually convey to you the chemical properties of the smells themselves. Apparently, the main function of primary olfactory cortex is actually sniffing. So they talk about this study in your textbook where people are either sniffing in when there's a smell present, or they're sniffing in when there's not a smell present. And they found basically similar amounts of activation in the olfactory cortex, primary smell cortex. It didn't matter whether there was a smell present or not. Just inhaling, that is sniffing, as though you were searching the environment for a smell, that will strongly activate primary olfactory cortex. So this seems to be a part of the brain, which is really important for sampling the environment, taking in information from the environment, using your motor system to kind of make your body do that however it does that, making your body sniff, inhale to sample chemicals from the environment. That seems to be the primary function of olfactory cortex, is actually making your body take in the chemicals. Whereas the secondary cortex, which is not shown here, the orbital frontal cortex. Do I have a picture right here? No, I don't. The orbital frontal cortex would be up here more towards the front of the brain. This seems to be a brain which responds more strongly when there is a smell present. When there's not a smell present, this region of the brain doesn't react as strongly, the orbital frontal, that is secondary smell cortex. But when there is a smell present, it does respond more strongly. Okay, so taste. I think this is the last. Oh no, we have taste and then a little bit of touch, but I'm not going to spend too much time on touch. So taste is another chemical sense, and it's has different types of receptors, just like smell does, though it appears to have less receptors than smell does. Smell has many different receptors. In the sense of taste, there usually are these 4 different receptors, which are kind of convenient, basically called, the salty receptor, the sweet receptor, the sour receptor, the bitter receptor. And these are located on different parts of the tongue. And each one responds differently to different types of chemicals. Those chemicals, which are salty, will cause one receptor to activate most strongly. For instance, this one right here, the fungiform, this responds most strongly to salty. It responds to all of the different chemicals, all the different tastes, but you can see here the yellow bar. Now it's not a good time for an update. It has a tall yellow bar right here, which suggests that this particular type of receptor responds most strongly to salty, whereas this type of receptor responds most strongly to bitter, and apparently this one responds most strongly to bitter as well. But there's kind of a different set of biases. Each type of receptor responds most strongly to different types of tastes, and each receptor differs in their strengths and weaknesses as far as the different types of chemicals that they process, the different types of tastes that they can process. So some more information about taste. I think I have a good picture of the cranial nerves. All of these senses that we've been talking about involve the cranial nerves. So once again, the cranial nerves go usually from the brain stem or from the spinal cord down at the lowest parts of the brain. They innervate the face, and then the information from the nose or from the eyes or from the ears or from the mouth, all of that information is using cranial nerves, which I think of as kind of like wires that are in your face. And they take that information and then they convey it to the brain stem before it reaches the cortex. And so several cranial nerves have been suggested to be involved with taste. And each cranial nerve appears to represent different locations on the tongue. So there's some suggestions that the different cranial nerves might differ in the extent to which they process different aspects of the types of sensations that your tongue can have, such as pain, which my understanding that carbonation, which allows soft drinks to taste very nice, is processed in a way kind of analogous to pain. So it's like one of those hurts so good type of phenomenon, because carbonation is pleasant, yet it's kind of like our body processes it as though it's type of pain. Similarly, probably to spicy food. And then there's the different cranial nerves could process where on the tongue something is, so the location and the temperature of something which is on your tongue. So different information is available on the tongue, and it appears that different cranial nerves are processing the different aspects of the different information. The primary taste cortex is located very similarly or very close to primary smell cortex. It kind of makes sense because they're both taste and smell are both the chemical senses. And there's also these interactions between smell and taste. Like when you're sick and you can't smell, if your nose is clogged up, then you don't really taste very well either. So smell and taste clearly interact a lot. And so to me, at least, it's not surprising that in the brain, their primary sensory areas for smell and taste are basically right on top of each other in this intersection between the frontal and temporal lobes. And just like smell, taste connects to the orbital frontal cortex. So orbital frontal cortex is like secondary smell cortex, but it's also secondary taste cortex. So both smell and taste primary areas send signals to the same secondary area, which is orbital frontal cortex. And I think now is when I have a picture of orbital frontal. No, it's yeah. Oh, it's this article right here talks about orbital frontal cortex. Okay. So here is the pathways right here for taste. Here are all those cranial nerves I was just talking about, and you can see how the different cranial nerves are taking in information from different parts of the tongue. The different receptors are on different parts of the tongue as well. And so different information from different receptors, different information on the tongue is sent to these various cranial nerves, which then connects to the brain around here, the brainstem or the spinal cord. That information is sent up to the thalamus, because we're talking about taste now. And then that information does go to the taste cortex, which is once again kind of on the boundary between the frontal and temporal lobes. The orbital frontal, this is a coronal section of the brain. So the orbital frontal part of the brain, which is the secondary taste area, it'd be cut off right here. So it's in the front of the brain. In this picture, they cut off the person's front of the brain. So the orbital frontal cortex would be towards the front of the brain right here, but it was cut off. So you can't see from here, from primary taste cortex, information will be going towards the front of the brain to that orbital frontal area, which is the secondary taste cortex, which is what I'm talking about here, orbital frontal cortex. I talked about this way at the beginning of this chapter. It's actually a secondary area for like all of the senses, at least from the research that I've read. The orbital frontal cortex it takes signals from each of the 5 primary sensory areas: primary visual, primary auditory, smell, taste, touch. All of the different sensory areas, the primary sensory areas, send signals to this frontal lobe region, the orbital frontal cortex. And I know at the beginning of chapter, I definitely showed you some pictures anatomically of where this is in the frontal lobe of the brain. And so one of the functions of this is presumably of the orbital frontal cortex is to integrate sensory information from all of the senses. And then, yeah, this is a study published in a journal, Brain and Cognition, a decent journal, about the presumed functions of orbital frontal cortex. And they make the argument here that it's important for learning the relationships, the orbital frontal cortex, kind of giving emotional strength or emotional valence to things based on their sensory properties. So what they call this, they call it stimulus reinforcement association learning. So you learn that a given stimulus, which would be, a given smell or taste or sound, or the combinations of, what a thing looks like and what it tastes like, or what a thing looks like and the sound that it makes, all of those different sensory things about a given object can cause you to have different emotional responses to that thing. If something tastes bad, then you're going to have a negative response to it. If it tastes good, you'll have a positive response. If it's a song that you dislike, then you'll have a negative response. If it's a song you really like, you'll have a positive response. Those are all kind of our emotional responses that we've acquired based on the sensory experience of something. And presumably the orbital frontal cortex is important for forming those memories, those stimulus response association learning, those memories that are formed based on our sensory experience with things. And then finally, somatosensation. I'm not going to really talk too much about this one. I guess I will mention that we have talked about the primary sensory cortex for this again. That is the somatosensory cortex, which is located in the parietal lobe, which is just posterior to the central sulcus. Central sulcus is the dividing line between the frontal and parietal lobes. Somatosensory cortex is located just posterior to that in the parietal lobe. And then we have these different receptors right here, and these different receptors process different pieces of information. So this is kind of a theme in all of these different senses. This is the last sense that we're going to talk about, is there's oftentimes different types of receptors, or at least receptors that are located in different parts of our body. Like for instance, in the visual system, the cones are located on the fovea, while the rods are on the peripheral parts of the retina. And those 2 different types of receptors process different types of information. And that happens in basically every sense, that the different types of receptors, or at least the location of receptors, varies the type of information which those receptors process. Similar type of thing for the sense of touch or somatosensation. And I'm not going to explain this because I think it's in your textbook, and I'm not going to really add too much other than the textbook, but there are these different receptors right here. They're located in different parts of the skin, and they process different types of information. So usually I do ask one question about this on the test, just making sure that you understand this basic concept. I'm not going to talk about it too much because I think you can do it by yourself, but there'll be a question basically about what are the functions of these different receptors and where are they potentially located in the skin. And that's where I'm going to leave for today, because then we get back to some of the big picture things, and we'll wrap up this chapter next time, I believe. So that's it. Call it a day here. So I'll see you Thursday.
Thank you.
Oh, you're welcome. See you later.